Ciao mondo, hello world, hola mundo, hello mon, konnichiwa, ni hao, namaste. We are here in India, in Hyderabad, and we had the chance to have a wonderful advisory board organized by Jaffron on hemoadsorption in different situations. And I am fortunate to be here with Professor Ansari, and we share the chairmanship of this advisory board where many doctors, intensivists, nephrologists and pediatricians from India gathered together to try to focus on the future research and the practice of hemoadsorption. So can you just summarize a little bit the content of our...
So, Professor, it's always a pleasure to bask in your glory. You have been the founding member of blood purification, but today it was a pleasure to dwell upon the three key areas which bother us: sepsis, liver failure, and poisoning, and the role of all forms of blood purification, especially hemoadsorption therapies. And it was a pleasure to have this interactive session, all the experts from India, and hearing from you and knowing the past and the future. Thank you so much.
So the main poisoning here is pesticides, right? Or snake bites.
Paraquat or snake venom, zinc phosphide, aluminum phosphide, the rat poisons.
Do you have a lot of cases here?
Not, unfortunately a lot. This is a farming-based country, so we have a lot of agricultural people, not the best conditions for them, and most of the people take easy way out to consume these poisons. Unfortunately, it's an unregulated market, okay, and has unprecedented effect with poor antidote penetration. In fact,
so you have a unique chance actually to study this type of pathology, because in other countries this is not so frequent. The other thing is today it was clear that both in sepsis and liver failure, the idea that hemoadsorption or DPMAS, the double molecular absorption system, can be used to gain time before you have transplant, surgery, or recovery from organ failure, right?
As you rightly said, the gain of time function can act as a bridge where none exists. And probably we can look forward where there was absolute darkness. We can have better therapies or definitive therapies to come in. And I love the way you explained about endophenotypes and how to kind of dive deep and look at these individual phenotypes.
So I think that one of the messages that emerged clearly is that we should start from daily operations to collect data and observe what are the characteristics of our patient and the outcomes in order to answer the unanswered question: when to start, which patient, how frequently. But before to do any study, we need to analyze what we have done so far, and you have a unique possibility because you have a large population.
So you rightly said the dose-drug conundrum which we always face: how long to give, how many filters to use, what should be the end point. And you so rightly said that don't just look at one simple biochemical endpoint. Look at biological endpoints, look at clinical endpoints, severity score. And I think it was really one of the best take-homes of this advisory board.
The other secret is you have to use a therapy in order to better understand it and to make sure that you use that therapy at the best. Otherwise, you have the risk that nurses are not well trained, doctors do not have a clear understanding. So there should be a parallel use of the technique and the other side preparing for a nice study, I think.
Absolutely. We have to go this learning curve chasm by getting every people together in a structured way, and who better to teach than you?
Well, we expect great actions and results from India. And with this, thank you very much for joining today, Cappuccino. We hope to see many Indian friends at the Vicenza course in June this year. And with this, have a wonderful day. Excellent.
Excellent.